डर होना चाहिए और वो दिल में होना चाहिए, और वो दिल आपको उनका नए सामने वाले का होना चाहिए। May I come in? वो क्या इतना बड़ा?
Welcome back, my dear AS students, to the ASRARIA notes coaching classes. Now we are going to study the ASRARIA notes RRR AL Rapid Revision AS series for the NEET UG Biology 2024. Now we are going to discuss about the topmost 100 AS topics in the real rapid revision, as it is real rapid revision. So the class will be about high yield AS topics, which can be there in your final exam, right? So let's begin with the first AS topic, Ernst Mayr. Ernst Mayr, born in 1904, died in 2004. He lived for 100 years, right? And Ernst Mayr is considered as the Darwin of the 20th century. You see, 100 runs in cricket is known as century. Ernst Mayr is considered as the Darwin of the 20th century, right? So Ernst Mayr got awarded 3 prizes in his career known as Triple Crown of Biology. The mnemonic to remember in the chronological order is Board of International Cricket. As 100 runs in the cricket is known as century, each and everybody is correlated. He lived for 100 years. He is considered as the Darwin of the 20th century, and he got Triple Crown of Biology. Chronological order, Board of International Cricket. B for Mason Prize in 1983. I for International Prize for Biology in 1994. And C for Crawford Prize in 1999. And Ernst Mayr pioneered the currently accepted definition of biological species. Means Ernst Mayr defined biological species. So Ernst Mayr is considered as the Darwin of the 20th century. Ernst Mayr defined biological species. Ernst Mayr got awarded Triple Crown of Biology. Characteristics of living organisms, right? There are non-defining features and defining features, right? Non-defining features are growth and reproduction, whereas the defining features of living organisms are cellular organization, metabolism, consciousness, right? Growth, as we know, increase in mass or size and increase in number of individuals are twin characteristics of growth. As we know, growth in the living organisms is from inside by cell division, but whereas growth in the non-living organism is by accretion. Whereas reproduction, we can see budding. Budding can be seen in sponges, hydra, and yeast, right? Budding can be seen in sponges, hydra, and yeast. S for sponges, H for hydra, and Y for yeast. So the mnemonic is our body is shy. What is the mnemonic to remember the examples of body? Our body is shy, right? True regeneration capacity can be seen in planaria. What is planaria? Planaria is a flatworm. So now the current situation of our national media, they can go up to any extent for the sake of TRP. The mnemonic to remember is TRP. True regeneration capacity can be seen in planaria. TRP. Right? Where we can see the fragmentation. Fragmentation can be seen in fungi, filamentous algae, and protonemamosis. In the unicellular organisms, such as bacteria, amoeba, and unicellular algae, reproduction and growth is synonymous, right? They are both inclusive events. We can see the assertion and reason questions here. If you get assertion, growth cannot be taken as defining feature of living organism. The reason is because non-living organisms also grows by accretion, accumulation of materials on the surface, right? Assertion, reproduction cannot be taken as the defining feature of living organisms. Reason, mules, hennes, sterile worker, honeybees, and infertile women couples do not reproduce. Now, defining features of living organisms, cellular organization, metabolism, and consciousness. Cellular organization is the defining property of life forms. Biological organization starts with submicroscopic molecular level or cellular level, right? Now, metabolism. What is metabolism? The sum total of all the biochemical reactions occurring inside the body of living organisms is known as metabolism. Metabolism is defining feature of all living organisms without exception. Metabolism is nothing but anabolism. Anabolism is a constructive process, example photosynthesis. Catabolism, destructive process, example respiration. Consciousness. Consciousness is the defining property of living organisms. Consciousness is the most obvious and technically complicated feature. Consciousness is nothing but sensitivity and irritability. Sensitivity means ability to sense the external or internal environmental stimuli. Irritability means ability to respond to the environmental stimuli, either physical, chemical, or biological. Only human being is the living organisms who has self-consciousness. So this is all about your characteristics of living organism. The number of species that are known and described range between 1.7 to 1.8 million, that is 17 to 18 lakhs, and this is known as biodiversity. Systematics. What is systematics? It is derived from the Latin word systema, means systematic arrangement of living organisms. Systematics includes taxonomy and evolutionary relationships. Taxonomy is the key part of systematics. There are 4 basic processes to taxonomy. Those are characterization, identification. Identification can be done directly with the help of herbarium and indirectly with the help of taxonomic key. Then third process is nomenclature, binomial nomenclature or binomial nomenclature. These binomial or binomial nomenclature was popularized by Carolus von Linnaeus. Linnaeus popularized binomial nomenclature. Linnaeus is considered as the father of taxonomy. Linnaeus is considered as the founder of modern systematics. And Linnaeus proposed 2 kingdom classification, Plantae, Animalia. And the 4th process is classification, right? So taxonomical classification forms taxonomical hierarchy, in which there are 7 obligatory taxonomic categories. What are they? There are 7 obligatory taxonomic categories: kingdom, division or phylum, class, order, family, genus and species, right? So Linnaeus, Carolus von Linnaeus popularized binomial nomenclature, father of taxonomy, founder of modern systematics, and 2 kingdom classification, Plantae and Animalia. Yes, my dear students, the process of naming an organism, plant or animal with a standard universally accepted scientific or biological name under the guidelines provided by ICBN. ICBN, I for international, C for code, B for botanical and N for nomenclature. ICBN, International Code for Botanical Nomenclature for Plants, or ICZN. I for international, C for code, Z for zoology and N for nomenclature. ICZN, International Code for Zoological Nomenclature for Animals, is known as nomenclature. The process of naming an organism, plant or animal with a standard universally accepted scientific or biological name under the guidelines provided by ICBN for plants, International Code for Botanical Nomenclature, ICZN for animals, International Code for Zoological Nomenclature, is called as nomenclature. If that scientific name consists of 2 words or 2 components known as binomen, then it is called as binomial or binomial nomenclature. Who popularized binomial nomenclature? This binomial or binomial nomenclature was popularized by Carolus von Linnaeus, right? What are the rules and guidelines provided by the ICBN? Let us see. Scientific name should be in Latin origin. Which origin? Latin origin. Why beta? Because Latin is a dead language. It cannot be changed, right? And printed in italic form. Which form? Printed in italic form to denote the Latin origin. Scientific name should be in Latin origin and printed in italic form. The 2 words of the binomial nomenclature, binomen. The first word is the genus, generic name is a noun and it always starts with a capital letter. The second word of the scientific name, species, specific epithet, is an adjective and always starts with a small letter. Example, scientific name of the mango is Mangifera indica. You see, Mangifera is the generic name, starts with a capital M. Indica is the specific epithet, starts with a small l, right? When handwritten, both the generic name and specific epithet are separately underlined. Example, Mangifera indica. You can see Mangifera is the generic name starting with the capital M is separately underlined. Indica is the specific epithet starting with the small I is separately underlined. Name of the scientist or author who described the plant species for the first time or who discovered the plant species for the first time and named the plant species for the first time is written in abbreviated form right after the specific epithet. That is at the end of the scientific or biological name. Example, Mangifera indica Lin. Mangifera is the generic name with the capital M is separately underlined. Indica is the specific epithet with small I is separately underlined. This species of mango, Mangifera indica, is first time described or discovered and named by the Linnaeus. Hence the name in an abbreviated form, Lin, after the specific epithet, right? Mangifera indica Lin. This is all about your rules and guidelines by ICBN. What is tautonymy? Tautonymy is applicable only in animals, zoology, not in botany, right? The process of naming an animal with a scientific name consisting of same genus and same species. Name is known as tautonymy. Naja, Naja, the Indian cobra. Axis, Axis, the spotted deer. Catla, Catla, Catla is the fish, right? It is applicable only to the animals, but not in the plants, right? We cannot say Parus, Parus for apple. We can say Parus Malleus, right? So tautonymy is only applicable for animals, not for plants. Right? The process of naming an animal with a scientific name consisting of genus and species which are same. Naja, Naja, Indian cobra, Axis, Axis, spotted deer, Catla, Catla, Catla. So this is all about the binomial nomenclature. Yes, my dear students, now we are going to study about the 7 obligatory taxonomical categories in the taxonomical hierarchy. Each category is a taxon at different levels, represents a rank and is a unit of classification, right? So these are the 7 obligatory taxonomical categories. What are they? Kingdom, division, phylum, class, order, family, genus and species. There are other categories also. Below the kingdom we can see subkingdom, right? Below the division of phylum we can see subdivision or subphylum, right? Above the class we can see superclass and below the class we can see subclass. Right, family, above the family you can see superfamily, below the family you can see subfamily. So there are other categories also, but there are only 7 obligatory taxonomical categories in the taxonomical hierarchy. Must. How to remember those 7 obligatory taxonomical categories? The mnemonic is King's Deficios class of genus species. King of Saudi Arabia invited all the genus species of the world to his kingdom for the feast, and he has taken a group photo selfie. And he kept that selfie photo as his DP of the social media account. So King's DP shows the class of genus species. King's, kingdom, D for division for plants, P for phylum for animals. Shows class, class of, O for order, F for family, genus, species, genus, generic name and species specific epithet. Kingdom is the highest category of the largest taxon. Species is the lowest category of the smallest taxon. According to Linnaeus, species is a basic unit of classification. Definition of species. Species is a group of morphological similar organisms sharing a common genetic pool which interbreeds freely and produces fertile offsprings. This biological concept of species given by Buffon, right? As we go higher from species to kingdom in the ascending order, common characteristics goes on decreasing. So up to what category level we can see the similarities of characteristics? Up to family level you can see the similarity of characteristics. From the order onwards to the kingdom we can see aggregation of characteristics. So this is all about your diversity in the living world first chapter. Let us see the next slide with the next chapter, biological classification. Yes, my dear AS students, welcome back to the ASRARIA Notes Real Rapid Revision AS series for NEET UG 2024. Let us start the biological classification chapter. Aristotle was the earliest scientist who classified plants into herbs, shrubs, trees, and animals with RBCs and animals without RBCs. Linnaeus has proposed 2 kingdoms classification, that is Plantae, plant kingdom, Animalia, animal kingdom. Haeckel has proposed 3 kingdoms classification, this Plantae, plant kingdom, Animalia, animal kingdom, Protista contains protist. Copeland has proposed 4 kingdoms classification, Plantae, Animalia, Protista and Monera. R.H. Whittaker proposed 5 kingdoms classification: Monera, Protista, Fungi, Plantae, Animalia. Kingdom Monera consists of unicellular prokaryotes. Prokaryotes are unicellular, and the sole member of Kingdom Monera are bacteria. Protista, Fungi, Plantae, Animalia are eukaryotes. Protista are eukaryotes but unicellular organisms. Fungi, Plantae, Animalia are eukaryotes with multicellular organisms. Right? Main criteria taken by the R.H. Whittaker for his 5 kingdoms classification is cell structure, thallus body organization, mode of nutrition, reproduction, phylogenetic relationships. Monera, Protista, Fungi, Plantae, Animalia. 5 kingdoms, 5 main criteria: cell structure, thallus body organization, mode of nutrition, reproduction, and phylogenetic relationships. Carl Bush and his co-workers proposed 6 kingdoms classification, leading to the formation of 3 domains: archaea, consisting of archaea bacteria, bacteria consist of eubacteria, and eukarya consisting of the eukaryotes. Domain is a highest taxon than kingdom. Carl Bush has proposed 6 kingdoms classification based on studies with the help of small unit of ribosomal RNA, 16S rRNA. So this is all about the classification. You can get them as the following one: Linnaeus proposed 2 kingdoms classification, Haeckel proposed 3 kingdoms classification, Copeland proposed 4 kingdoms classification, R.H. Whittaker proposed 5 kingdom classification. Or you can get one-liner: who proposed the 5 kingdom classification? R.H. Whittaker. Or you can get this statement wise: what are the following are the main criteria for the R.H. Whittaker 5 kingdoms classification? Right? Let us begin with the kingdom Monera. Kingdom Monera consists of prokaryotes. That is, true nucleus or nuclear membranes are absent. Instead of true nucleus, we have nucleoids or genome, naked circular DNA. They are prokaryotes and unicellular single cell organisms. Bacteria are the sole members of Monera. Monera is divided into 2: archaebacteria, eubacteria. Eubacteria, nothing but true bacteria. Eu means true, is nothing but true bacteria, right? Archaebacteria, assertion, can survive in extreme conditions. Reason: their cell wall composition is different from the eubacteria. That is, this shows pseudomurine, right? How many types of archaebacteria we have? Halophiles, they live in salty areas. Thermoacidophiles, thermo means temperature, they live in hot springs. Methanogens, methane, right? They are present in the gut of ruminants of cows, buffaloes. They produces methane gas or biogas from the dung of cows and buffaloes, right? Now, let us see the eubacteria. True bacteria, right? True bacteria. They shows cell wall, right? Which is made up of murine or mucopeptide or peptidoglycan. These many names. Cell wall of the bacteria is made up of murine, mucopeptide or peptidoglycan. Eubacteria is divided into 2: autotrophic bacteria, heterotrophic bacteria. Autotrophic means they can synthesize their own food. Heterotrophic, they cannot synthesize their own food. They depends on the host. Heterotrophic eubacteria are the most abundant bacteria present, right? Majorly, they are decomposers. So, some uses of heterotrophic bacteria, let us see. Making curd from milk. दूध से दही बन जाता अपने घर में. Why? Because of the presence of lactic acid bacillus, LAB. Next, production of antibiotics, right? Antibiotics can be produced with the help of bacteria. Example, streptomyces. And the third one is fixing nitrogen in the legume roots, right? Example is rhizobium. Done. Now, autotrophic bacteria are of 2 types: photosynthetic autotrophs, chemosynthetic autotrophs. Photosynthetic autotrophs, examples are cyanobacteria, blue-green algae, consist of what? Chlorophyll, chlorophyll A, similar to the plants. Examples of cyanobacteria, Anabaena and nostoc. How can you remember the example of cyanobacteria? C for cyanobacteria, A for Anabaena, and N for nostoc. So can, C-A-N, can, right? In the nostoc, we have heterocyst, specialized cells fixing atmospheric nitrogen. right? In the nostoc, we have heterocyst, specialized cells fixing atmospheric nitrogen. While you are watching the real rapid revision ASRARIA notes AS series, you have to take NCERT textbook along with you so that you can compare it with the diagrams given in the textbook, right? In the nostoc, we have heterocyst, specialized cells fixing atmospheric nitrogen, and the colonies are generally surrounded by gelatinous sheet. Based on the shades, bacteria are of four types. If the bacteria are spherical in shape, then they are known as cocci, singular coccus. If they are rod shaped, then the bacteria are known as bacilli, singular bacillus. If they are comma shaped, then the bacteria are known as Vibrio. If they are spirally coiled, spiral shaped, then the bacteria are known as Spirilla, right? अभी अपन पढ़े LAB lactic acid bacillus, तो example of bacillus lactic acid bacillus हो गया. Cocci, Diplococcus, Pneumococcus, right? Staphylococcus, Streptococcus, comma shaped Vibrio cholerae, spirally coiled, spiral shaped Spirilla, Spirochete. So these are the shapes of bacteria. Let us proceed with the structure of prokaryotic cell, that is bacteria. Prokaryotic cell means a cell which lacks true nucleus, means nuclear membranes are absent. Instead of true nucleus, we have nucleoid, circular naked DNA chromatin material, also known as genome. In addition to these naked DNA chromatin material, some small circular extra genomic DNA are present in the bacteria, known as plasmid, right? Plasmid are very much useful, right? It gives the unique features to the bacteria, such as resistance to the antibiotics action, and it is used to monitor the bacterial transformation with foreign DNA. And plasmids are used as vector in rDNA technology, right? TI plasmid, PBR 322, you study, right? All the cell organelles are absent in the prokaryotic or bacterial cell except 70S ribosomes, 50S and 30S join with the magnesium. That S is Swedberg unit, indirectly measures the density and size of ribosomes. Then, along with the 70S ribosomes, there are some inclusion bodies present in the cytoplasm of the prokaryotic cell bacteria. Those inclusion bodies are the reserved food materials, such as phosphate granule, glycogen granule, cyanophysin granules. These are the what? Granules. Phosphate granules, glycogen granules, and cyanophysin granules. And gas vacuoles are present in blue-green or purple or green photosynthetic bacteria. These innermost plasma membrane is similar to the plasma membrane, cell membrane of eukaryotes. They shows invagination in the bacterial cytoplasm. That invagination of the plasma membrane or cell membrane is known as mesosomes. What is the function of mesosomes? Mesosomes helps in cell wall formation, DNA replication, and they helps in respiration and secretion, etc. Surrounding the cell membrane, we have cell wall. Cell wall of the bacteria is made up of peptidoglycan or mycopeptide or murein. If we stain the bacteria with a gram staining, then we get two types of bacteria. Gram-positive bacteria appearing blue-purple color, gram-negative bacteria appearing pink-red color. Why? Because of the presence of teichoic acid in the cell wall of gram-positive bacteria. Surrounding the cell wall, we have another outermost protective layer known as glycocalyx. Right? If it is loosely arranged, then it is known as slime layer. Loosely L, slime L. Loosely arranged, then it is known as slime layer. If glycocalyx is thick and tough, it is tightly bound, then it is known as capsid or capsule. Done? So this cell membrane, cell wall, and glycocalyx togetherly form cell envelope. What is the function of cell wall or cell envelope of the bacteria? It determines the shape, provides structural support, and prevents desiccation or bursting of the bacteria. So this. Now we can see from the cell wall, we get flagella, motile, right, structure. With the help of flagella, bacteria shows motility. Flagella helps in motility, right? From the cell wall, we get another structure such as villi, which is made up of protein known as fillin. Some of these villi form the sex villi, which helps in the formation of the conjugation tube in between two bacteria which undergoing conjugation, right? Conjugation is discovered by, right, Liderberg and Tatum in E. coli. Another structure coming out from the cell wall is fimbriae, and these fimbriae helps in attachment of bacteria to the substratum or host cells. So this is all about your prokaryotic cell or bacterial cell. Mycoplasma, PPLOs, pleuropneumonia-like organisms. Mycoplasma are the smallest living cell known without cell wall. They completely lack the cell wall and they can survive without oxygen, right? Smallest living cell with cell wall, bacteria. Do not confuse the question. If you get the question, smallest living cell known is Mycoplasma, or without cell wall is Mycoplasma, because they completely lack the cell wall. But whereas if the question is smallest living cell with cell wall is, your answer should be bacteria. Let us begin with the kingdom Protista, right? Protista kingdom consists of eukaryotes, the two unicellular single cell organisms, eukaryotes. Eukaryotes means presence of true nucleus, right? Presence of true nucleus means nuclear chromatin material surrounded by nuclear membranes, right? I think you have heard about the song Despacito, right? The mnemonic to remember the kingdom Protista is Despacito. D for Dinoflagellates, E for Euglenites, S for slime molds, P for protozoans, and C for Chrysophytes. Let us begin with the Dinoflagellates, right? Dinoflagellates. नाम में ही है dino means दो flagella. Cell walls are made up of cellulosic plates. In between those cellulosic plates, rows shows two flagella. Longitudinal flagella, horizontal flagella, right? Dinoflagellates, cell wall is made up of cellulosic plates which shows two flagellae, longitudinal flagellae and horizontal flagellae. Red dinoflagellates, that is goniolites, undergoes rapid multiplication which is responsible for the formation of red tides in the Mediterranean Sea. And these dinoflagellates releases toxins which kills fishes. Okay? So, important point here is goniolots, red dinoflagellates, undergoes rapid multiplication, right? Red tides in the Mediterranean Sea. Let us go for the D, euglenoids. Example, euglena, which shows longitudinal penile fission, right? They shows chromatophores consisting of chloroplast performing photosynthesis. They lack cell wall. Euglenoids lack cell wall. Instead of the cell wall, they have protein-rich layer known as pellicle. They have protein-rich layer known as pellicle. And euglena shows two flagella, right? One long and one short. Euglena shows two flagellae, one long and one short. But whereas dinoflagellate shows longitudinal and horizontal. Then the important point here is euglenoids lacks cell wall. Instead of cell wall, they shows protein-rich pellicle layer. Next, S for slime molds. Slime molds are the saprophytic protist. During the favorable condition, they form plasmodium aggregation, and during unfavorable condition, they form fruiting bodies from the plasmodium. Protozoans. Protozoans lacks cell wall, right? All protozoans are heterotrophs. Example of protozoans are AFCS, Armed Force College of Science. A for amoeboid. Parasite shows pseudopodia, example entamoeba, right? Then F for flagellated protozoans. Example, trypanosoma causes sleeping sickness and the vector is cystic. Very much important point here. So important point here to remember is flagellated protozoan, trypanosoma gambiense causes sleeping sickness and the vector which carries this trypanosoma cruzi gambiense is cystic. C for ciliated protozoans, example paramecium, they show cilia, peritrichus, and they show cavity, that is gullet. And S for sporozoans, the most notorious sporozoan is Plasmodium malarial parasite. The most common plasmodium is Plasmodium vivax, right, which is causing benign malarial fever. And C for Chrysophytes. Chrysophytes consist of golden algae, also known as desmids, and diatoms. Diatoms are the chief producers of oceans, right? Diatoms are the chief producers of oceans. The cell wall of the diatoms shows outer epitheca, inner hypotheca, upper epitheca, lower hypotheca, and they gives soapbox appearance. Assertion: cell wall of the diatoms are indestructible. Reason: cell wall are embedded with the silica in the oceans. Diatomaceous earth Kieselguhr is used in polishing, filtration of syrups and oils. So this is all about your kingdom Protista. जैसे ही आपको kingdom Protista आया, आपको गाना याद आ जाना. Despacito. D for Dinoflagellates, E for Euglenoids, S for slime molds, P for protozoan, and C for Chrysophytes. Dinoflagellates के अंदर utmost important क्या है? Goniologs, red dinoflagellate undergoes rapid multiplication responsible for the formation of red tides in the Mediterranean Sea. Euglenoids के अंदर important point क्या है? Euglenoids lacks the cell wall. Instead of that, protein-rich pellicle is present, right? Protozoans के अंदर important point क्या है? Flagellated protozoan, Trypanosoma, causes sleeping sickness, and the vector is CC, right? And whereas sporozoans, the most notorious is Plasmodium, malarial parasite. Plasmodium vivax is the most common one, causing benign malarial fever. And in the Chrysophytes, golden algae, desmids, and diatoms. Diatoms are the chief producers of oceans, and cell wall of the diatom shows upper epitheca, lower hypotheca, giving the soapbox appearance. Assertion: cell wall of diatoms are indestructible. Reason: cell wall is embedded with the silica. लोअर ओशन के नीचे कई मिलियंस ऑफ ईयर्स के अंदर जमा होकर के क्या फॉर्म होता हो? डायटोमेशियस अर्थ, आल्सो नोन एज कीसेलगुhr, that is used in polishing, filtration of syrups and oils. This is all about kingdom Protista. Let's begin with the kingdom fungi, right? Fungi are the heterotrophic thallophytes. Fungi are cosmopolitan in distribution. जहां बोले वहां अवेलेबल रहते हैं, जहां बोले वहां स्प्रेड हो जाते हैं हम लोग, right? Fungi forms are two forms. First one, yeast form, that is unicellular yeast form. Saccharomyces cerevisiae, which is used to make bread and beer, and Cryptococcus neoformis, which forms brain meningitis in human beings. Another one is multicellular molds form. Molds form means filamentous thallophytes, that is hyphae. The network of hyphae is known as mycelium, and that hyphae mycelium consists of multinucleated cytoplasm. Then it is known as cenocytic hyphae. Then dimorphic fungi. Two morphic morphology. They can be in the yeast form at 37 degrees Celsius and also in the molds form at 25 degrees Celsius. Those fungi known as dimorphic fungi. Very much important in MBBS second year microbiology subject, mycology. You are going to study about it there, right? Fungi cell wall is made up of chitin and polysaccharides, right? Fungi का cell wall किससे बने हुआ है बेटा? Chitin से बने हुआ है और polysaccharide. Chitin और कहाँ पे आया आपने को? Exoskeleton of cockroach is also made up of chitin. Sexual spores of the fungi are ascospores, basidiospores, oospores. अपने हैदराबाद में मारुति सुजुकी का कार डीलर है, राइट? साबू भाई, उस तरीके से निमोनिक याद रखो. S for sexual spores, A for ascospores, B for basidiospores, O for oospores of the fungi, right? As I told you, fungi is a cosmopolitan in nature. It is present everywhere, right? So fungi is divided into four classes: ascomycetes, basidiomycetes, phycomycetes, and imperfect deuteromycetes. The mnemonic to remember is ABP-ID. As we have our national television, ABP News, anchors will go anywhere by keeping their ID with themselves, right? Fungi जैसा जहां बोले वहां फैले हुआ है, कॉस्मोपोलिटन डिस्ट्रीब्यूशन है. अपने नेशनल टीवी एबीपी न्यूज के एंकर्स भी आईडी लेके जहां बोले वहां पहुंच जाते न्यूज देने. उस तरीके से इजी निमोनिक याद रख लो. डन. एस्कोमाइसिस, बेसिडियोमाइसिस, फाइकोमाइसिस, एबीपी, आईडी, imperfect fungi, that is deuteromycetes, right? चलो अपन सबसे पहले पढ़ेंगे फाइकोमाइसिट्स में. फाइकोमाइसिट्स को अपन बोल रहे हैं algal fungi, क्योंकि उसका स्ट्रक्चर सेम algae के जैसा रहता है. Phycomycetes, also known as algal fungi. In the phycomycetes, mycelium is aseptate and cenocetic. Very much important high yield AS point. Mycelium is aseptate and cenocetic in phycomycetes, that is algal fungi. Example क्या है बेटा? Rhizopus है, bread mold, bread के ऊपर फंगस जो आ जाता अपने. Albugo है, albugo is a parasitic fungus that causes white spots on the mustard leaves, right? And mucor. अब इसको निमोनिक कैसा याद रखेंगे आप? है मेरी परम 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 सुंदरी, गाना है ना, उस तरीके से निमोनिक याद रखना है. P for phycomycetes, A for algal fungi, R for rhizopus, A for albugo, M for mucor. So mnemonic is param, right? Phycomycetes, algal fungi, rhizopus, albugo and mucor. परम सुंदरी याद रखना है फाइकोमेसिट्स क्लास. ठीक है, चल अब आएंगे एस्कोमेसिट्स में. एस्कोमेसिट्स को क्या बोल रहे अपन? सैक फंगाई बोल रहे. नाम में से देखो एस्कोमेसिट्स के अंदर, ठीक है, SAC वहीं पे हो जा रहा, सैक फंगाई. डन. फिर एस्कोमेसिट्स में M है, M फॉर मोरेल्स. माइसिट्स में T है, T फॉर ट्रफल्स. तो मोरेल्स एंड ट्रफल्स आर edible in the ascomycetes. Mycelium is septate and branched. एग्जाम्पल क्या है? एस्परजिलस, एस्परजिलस नाइगर, ब्लैक फंगस, एस्परजिलस फ्यूमिगेटस, ग्रीन फंगस, राइट. सैक्रोमाइसिस अभी पढ़े अपन, ईस्ट यूनिसेलुलर फॉर्म में है, ईस्ट फॉर्म में सैक्रोमाइसिस ईस्ट यूनिसेलुलर. और क्या है? क्लाइविसेप्स. एस्कोमेसिस, ए फॉर एस्परजिलस, एस फॉर सैक्रोमाइसिस ईस्ट यूनिसेलुलर, सी फॉर क्लाइविसेप्स, राइट. Penicillium fungi is used for the production of penicillin antibiotic. Another important AS point here is Neurospora. Neurospora is the fungus belonging to the class Ascomycetes, right? It is used extensively in biochemical and genetic work. Very much important AS point. Done? हो गया. Ascomycetes नाम में छे. A for Aspergillus, S for Saccharomyces is unicellular, C for Claviceps, M for Morels, C for Truffles are edible, delicious. Ascomycetes are also known as sac fungi. SSC उल्टा नीचा कर दो. Mycelium is septate and branched. Penicillium is used for the production of antibiotic penicillin. Neurospora, very much important high yield AS point here is Neurospora, fungus belonging to the class Ascomyces, is used extensively in biochemical and genetic work. Done. Next class, Basidiomycetes. Basidiomycetes को क्या बोल रहे हैं अपन? क्लब फंगाई, राइट, और ब्रैकेट फंगाई. पफ बाल्स दिख रहे उसमें. Mycelium is septate and branched. एग्जाम्पल्स क्या है बेटा? एग्जाम्पल्स क्या है देखो? Agaricus mushroom, Ustiligo smut, Usinia rust causing wheat rust, right? 6 organs are absent in the Basidiomycetes. तो इसमें इंपॉर्टेंट पॉइंट क्या है? Usinia causing wheat rust is a parasite. Done. अब इसको निमोनिक कैसा याद रखते हैं आप? आपके हिसाब से याद रख लो. क्लब सी, Ustiligo smut, Usinia rust, wheat rust. तो कब? ठीक है, कब याद रख लो आप? 6 organs are absent in the Basidiomycetes. चलो नेक्स्ट वन, Deuteromycetes. लास्ट क्लास क्या है? Deuteromycetes. Deuteromycetes को अपन imperfect fungi बोल रहे हैं. Assertion, Deuteromycetes are also known as imperfect fungi. रीजन क्या है बेटा? Only vegetative और asexual phase is known. खाली vegetative और asexual phase मालूम है अपने. सेक्सुअल रहा तो उसको दूसरे क्लास में डालेंगे. Asexual spores of Deuteromycetes are nothing but conidia, right? Mycelium is septate and branched. देखो आप हर क्लास में देखो, Mycelium is septate and branched. Mycelium is septate and branched. Mycelium is septate and branched. सिर्फ एक Phycomycetes में कैसा है? Mycelium is aseptate and cenocetic. ये important point. ठीक है, चलिए Deuteromycetes क्या रहते बेटा? D for Deuteromycetes. D for mostly they are decomposers of litter and help in mineral cycling. Example क्या है बेटा? Alternaria, Coledotrichum, Trichoderma. अब इसको कैसा निमोनिक याद रखते हैं आप? Deuteromycetes का एक्ट. तो आज के प्रेजेंट जनरेशन, प्रेजेंट टाइम में एंकर्स ऑफ द न्यूज एंकर्स ऑफ द न्यूज चैनल्स, दे आर नॉट एट ऑल रिमेन्ड एज एंकर्स. नाउ दे आर एक्टर्स, तो उनको एक्ट करना पड़ता, राइट। निमोनिक क्या बोले थे अपन, एबीपी आईडी बोले थे, नहीं, एस्कोमाइसिट्स, बेसिडियोमाइसिट्स, फाइकोमाइसिट्स, एबीपी न्यूज जहां बोले वहां पहुंच जाते हैं आईडी दिखाने। इम्परफेक्ट फंगाई डिटेरोमाइसिट्स, राइट। अब डिटेरोमाइसिट्स इम्परफेक्ट फंग, उन लोग इम्परफेक्ट है, एंकर्स तो रहे नहीं, एक्टर्स हो गए, तो लोग क्या कर रहे हैं, एक्ट कर रहे। अल्टरनेरिया, कोलेटोट्राइकम, ट्राइकोडर्मा। सो दिस इज ऑल अबाउट योर किंगडम फंगाई। एटमोस्ट इंपॉर्टेंट आपके लिए यहाँ पे क्या है, फंगल सेल वॉल इज मेड अप ऑफ काइटिन, सेक्शुअल स्पोर्स और एस्कोस्पोर्स, बेसिडियोस्पोर्स, ओस्पोर्स, साबु निमोनिक, और उसकी क्लासेस क्या क्या है, फाइकोमेसिस, एस्कोमेसिस, बेसिडियोमेसिस, डिटेरोमेसिस। असर्शन, डिटेरोमेसिस नोन एज imperfect fungi. Reason, only vegetative resection phase is known, right? Phycomycetes, mycelium is aseptate and sinusotic. Mnemonic क्या है, परम सुंदरी, परम, P for phycomycetes, A for algal fungi, R for rhizopus, bread and mold, albigo, causing white spots on mustard leaves and mucor. But whereas in ascomycetes, in the name itself, A for Aspergillus, S for Saccharomyces is unicellular, C for Claviceps. Penicillium is used for the production of antibiotic penicillin. At most important point, Neurospora is used extensively in the biochemical and genetic world. Right? Basidiomyces also known as club fungi. Ustuligo smut, Pusinia rust, causing wheat rust. Cup is the pneumonic cells organs are absent, agaricus, mushroom. Right? Then dikaryophase, very much important high point, high yield AS topic is dikaryophase is seen in ascomycetes and basidiomycetes. एक ही सेल में 2 न्यूक्लियाई रहते हैं एक तरह, उसको बोलते हैं डाइकैरियोफेज, डाइकैरियन, क्या बोलते हैं उसको, डाइकैरियन, 2 न्यूक्लियस रहते हैं, तो डाइकैरियोफेज is seen in ascomycetes and basidiomycetes. So this is all about kingdom fungi. Algae and fungi show symbiotic association known as lichens. Algal component is known as phycobiont, autotroph. Fungal component is known as mycobiont, heterotroph. Lichens are very good pollution indicators. Resection. Reason, lichens do not grow in polluted areas. Another symbiotic association of fungi along with the roots of higher plants known as mycorrhizae. Virus is acellular. It is neither a living nor non-living, right? Virus is acellular, inert crystalline structure. Nucleoproteins, made up of nucleoproteins. Nucleic acid, that is infectious genetic material, either DNA or RNA, but never both. Protein coat known as capsid shows subunits known as capsomers, right? So viruses, acellular, inert crystalline structure, made up of nucleoproteins, nucleic acid and protein coat. Protein coat is known as capsid, subunits of capsid known as capsomers. Nucleic acid is an infectious genetic material, either DNA or RNA, but never both. Virus was discovered by Dmitry Ivanovsky. The name virus given by Martin William M.W. Vizernik, infectious living fluid, contagious vivum fluidum, and the virus was first crystallized by W.M. Stanley, shows largely proteins. Then, and the viruses that infects the plants have single-stranded RNA as the genetic material. Example, tobacco mosaic virus, TMV with the helical structure. And in the tobacco mosaic virus, we can see capsid shows subunits capsomers, and there are 2130 capsomers in the TMV. But whereas the virus that infects bacteria are known as bacteriophage, and they have double-stranded DNA as the genetic material. Then, chali. Now, viroids. Viroids are the smaller than viruses. Why? Because they are containing only nucleic acid with them, that too free single-stranded RNA of the low molecular weight, and they have no proteins with them. Viroids discovered by T.O. Diener in 1971. Right? Viroids causes potato spindle tuber disease. But whereas prions, no nucleic acid, but they have only protein coat, abnormally folded proteins, they causes BSE, bovine spongiform encephalopathy, that is mad cow disease in cattle, and similar variant of it is CR Jacob disease, CJD, in human beings, right? So this is all about your algae, fungi, and viruses. Bacterial diseases, some of the bacterial diseases given in your NCERT textbooks are citrus canker, cholera, typhoid, tetanus. Viral diseases are mumps, AIDS, herpes, chickenpox variety, influenza, smallpox. जब क्रिकेट दुनिया में महेंद्र सिंह धोनी MSD जब आकर के हेलीकॉप्टर शॉट्स मार करके अच्छे रन्स बना रहे थे, चेस कर रहे थे, बेस्ट फिनिशर कहलाए, राइट? तो क्या वायरल हुआ था? जब माही मार रहा है, माही मार रहा है, राइट? तो माही, वायरल डिजीजेस, माही, मम्प्स, एड्स, herpes, influenza. महेंद्र सिंह धोनी माही मार रहा है, माही, मम्प्स, एड्स, herpes, influenza, सिंह, स्मॉलपॉक्स, धोनी डिजीजेस, वायरल डिजीजेस, वो वायरल हुआ था. तो प्लान सिम्टम्स इन वायरल डिजीजेस क्या है बेटा? वही वायरल क्या हुआ था? महेंद्र सिंह धोनी MSD, मोजाइक फॉर्मेशन, स्टंटेड ग्रोथ, ड्वार्फिज्म, लीफ रोलिंग, curling, yellowing, and green clearing. So this is all about your bacteria and viruses. अगर आपको वीडियो अच्छी लगी रियल रैपिड रिवीजन की, तो मेरे वीडियोस को लाइक करिए, शेयर करिए और मेरे चैनल असरारिया नोट्स कोचिंग क्लासेस को सब्सक्राइब करिए. मिलते हैं अगले वीडियो में अगली AS टॉपिक रियल रैपिड रिवीजन क्लास के साथ. टिल देन टेक केयर.
Monster.